Thank you for having me. I am going to turn off my video because I have bandwidth issues sometimes. So thanks again, Joey, for having me and inviting me. When Joey invited me, he had mentioned that flowering rush seemed to be of particular interest. And so I thought I would really mostly focus today's presentation on that system. Before I begin, I think it's always helpful to start with what is biocontrol, and that's the intentional use of one living organism to control or suppress another. So in this case, we're talking about noxious weeds. And with my program, we're focused mostly on insects, some mites, some pathogens, and very rarely nematodes.
Hey, Jenna, this looks great, but you can go into presentation
Oh
mode.
gosh, I'm so sorry.
It's okay.
Thank you for
Still
pointing
looks
that
great.
out.
Yeah,
I
thank
can
you.
see it all. So just to give you a sense of my program, it is a statewide program in Washington. I am with Washington State University, the Integrated Weed Control Project Director. And this is providing biocontrol agents to land managers, and that really can be anybody who has appropriate sites for biocontrol. If you are interested in biocontrol agents and beyond flowering rush, other potential options for biocontrol, you're always welcome to contact me. If you're in other states and you're not sure who to talk to in your state, you can also contact me and I can see what we can do about getting you biocontrol agents or getting you in touch with the right people. I also provide education, and one thing that I think is really important is that we're providing expertise about biocontrol and how best to incorporate it into the overall weed management strategies of your program. As part of that, I am involved in several research projects, and today I'll be talking about flowering rush. So if you're not familiar with flowering rush, this is a freshwater aquatic invasive species. You can see the flower is quite distinct, but it rarely flowers out here in the West. It does colonize wetlands, slow-moving rivers, canals, and irrigation ditches. So we often see it in the back channels of rivers. That's where it tends to settle in and make larger populations. But we do see it in areas where the river is moving. So my feeling is you kind of always want to keep your eye out for this weed. In North America, we have two cytotypes, a diploid cytotype and a triploid. So the triploid is sterile, and this is the cytotype we see most often in Western North America. It does not produce seeds. It rarely flowers, so it's almost exclusively reproducing using rhizome buds or rhizome fragments, as you can see in this bottom right picture. The diploid cytotype is more dominant in Eastern North America, so around the Great Lakes and Midwest area. And this does produce by seed, but it also has bulbils on the rhizome and inflorescence. So it looks quite different. The pictures are fairly dramatic when you see the diploids, but you're unlikely to see those out here in the West. It has several different growth forms, and this makes it particularly difficult for management. It can grow fully emergent, as you can see in the bottom right picture. Partially submerged, and then we have a lot of populations in the West that are fully submerged. So this picture at the top right shows, this is a picture from Flathead Lake, where you can see there is emergent plants all around this, but the primary growth form at this site is fully submerged plants. And this makes it very difficult to detect, and it's very easy to overlook it. So oftentimes the populations are fairly well established before you even recognize this plant is there. It has a couple of pretty important impacts. It's an ecosystem engineer, and for us out in the West, it's particularly concerning because it's providing habitat for our non-native northern pike, which are feeding on our salmon. And so it's important for us to try to reduce that habitat for northern pike in order to help salmon. It is also habitat for the snail that carries swimmer's itch. So that's a pretty, if you've ever had swimmer's itch, that's a pretty important reason to not have the snails around. And then it also can impact fishing and whether people can access their docks for boating. And the cost of control can be pretty high. So there's been a lot of different activities trying to figure out how best to control flowering rush. There's a lot of studies on herbicide use, which have been effective in some places, but are not widely effective. You can see the picture on the bottom left of covering the plant for 3 years. You pull off the cover, the plant's still growing. There's pretty labor-intensive work like hand pulling and diver-assisted suction. The challenge with that is that if you miss any of the pieces or you break the plant up at all, you could be spreading your population downstream. And then down in southern Idaho in the irrigation ditches, they've actually created a special bucket just for removing flowering rush out of those irrigation ditches, which can be pretty pricey work. So although there are all these different techniques, controlling flowering rush is really challenging. And it seems that site conditions have a really strong influence on our successes. This is the distribution of flowering rush in North America. It's found in the temperate zone. You can see on the eastern side of the continent, it really tends to cluster around the Great Lakes in provinces and states around there. In the west, we have it from about Edmonton south. Our most southern population that we know of is in Colorado, but it is most dominant in Alberta, Montana, Idaho, Washington, and Oregon has a few populations. More close to home this is what the distribution looks like within the Columbia Basin. So you're probably very familiar with the Columbia Basin being such an important water shed for us. And so there's a really high priority for trying to manage flowering rush within this region. On this map, you can see that all the red areas are where it's been found. Green represents where they've surveyed, but it isn't present. But we do tend to see that these every year, that red keeps on spreading. So it's because flowering rush was so challenging and we could kind of see the writing on the wall of this is a plant we're going to have a really hard time controlling. What are our management options? And so for that reason, we started to explore the potential of biocontrol. And we formed a consortium in 2013. I am the consortium lead for this project. And it's a partnership between CABI Switzerland, which is a research group that works on biocontrol development. And then it was also involved several states and provinces. One of the reasons we were excited about it is because it's the only species in the Butamaceae family. So this is increasing the likelihood we're going to find something host specific. So it's an important potential tool. And at that point, we had to start looking for funding opportunities and partners. We have a lot of different partners. This isn't even all of them, but these are sort of the major players. So I want to be sure to acknowledge that this is not work that I am specifically doing. It's part of a large group coming together to be able to make this possible. I'll just briefly cover the genotypes. There are two different genotypes that are dominant genotypes in North America. And the reason we needed to look at genotypes was because it helps us find better biocontrol agents in the plant's native range, which is Europe. So in the West, we have genotype 1, which is represented in blue. You can see that's fairly widely distributed. This does tend to be the triploid. And then in the East, we have more dominant genotype 4, and this is the diploid. And this is the sort of matching that we're trying to do, looking at European versus North American flowering rush populations. So anything that's colored circle is a genotype found in North America, and anything that's just a black symbol, those are all European sites. And what you can see is that we have a really hard time finding anything that's closely related genotype-wise to our genotype 1 in Europe. So this has made work quite challenging, but you can see at some point we did find a site in the Netherlands, and this was really important for our work on one of the biocontrol agents. So there are 3 different biocontrol agents we've been looking at, and I'm going to run through each of them, and I'll start with the one that still needs the most work and end on the one we're furthest along on. So the one that is kind of furthest away from being possibly released is a white smut, Dosantia nislii. And we're pretty excited about this. It has the potential for high damage. Other biocontrol systems have used white smut with great success. It's adapted to the aquatic environment. And one of the reasons we really want to focus on this is because it's able to infect plants that are fully submerged. And as I mentioned, we have a lot of those populations here in the West where we have fully submerged plants. So the insect biocontrol or potential biocontrol agents may not do so well in a fully submerged situation. So we really need something that's going to go after those populations. It's been quite challenging to find a strain of the smut that will attack our genotype. And so I mentioned the Netherlands. We did find a match between the Netherlands and our genotype 1, but unfortunately we didn't find any smut at that site. So 2 years ago, year and a half ago, they were able to find a strain in Romania that will infect our genotype 1 and 4. So this is pretty exciting. It's this is sort of a huge next step for us. Unfortunately, pathogens are really complicated. Their life cycles are very difficult. And so there's a lot more work that needs to be done on how to inoculate and test this species. The second potential biocontrol agent is a fly, Phytolyromyza ornata. This is, at first we weren't too excited about this fly because it just mines, the larvae are mining the leaves and the flowering stems. And we really wanted something to go after those roots. But after studying it a little bit more, our excitement started to grow. Plants began wilting within 2 to 3 weeks after exposure to a single female. You can see those pictures in the bottom left. You can see what that wilt looks like. And some impact studies have demonstrated that within 2.5 months, you can have a 37% biomass reduction, and that increases to 68% after 4 months. So those are great. Those are great sort of numbers that we're looking for. The other great thing about this fly is that it appears to be incredibly host specific. So it's been tested on 40 different test plant species and hasn't developed on any of them. So we're almost done testing on this. There's just a few more species this summer. And this fall, we will be writing the petition for field release, which will then go into both Canada and the US for review. So the last one I wanted to touch briefly on is Bagus nodulosus. This is the one we're furthest along on, and we hope to be doing releases soon. The larvae are developing in the leaves in the top part of the rhizomes. And what's interesting is the larvae are able to leave the plants and they swim to other plants. And so we're not sure exactly how that's going to influence things, but it's kind of a funky characteristic of a weevil. So it's gone through a lot of host specificity testing between 2016 and 22, and 45 different test plant species have been tested. So a pretty broad range considering that flowering rush doesn't have any close relatives. We found that the adults were laying no eggs on the plants. We never found any complete development, meaning we were never able to go from an egg to an adult on any of the test plants. And we also saw very minimal feeding even on our non-target species. So very host specific, a really clean looking biocontrol agent. It also impacts the plant pretty aggressively. We see it actually kind of hits it in a 2-phase response. So initially, the leaves start to die back and we see that the above ground biomass is reduced. But then later in the season, what happens is the plant then reallocates its resources and puts a lot more growth into the above ground biomass, and that is decreasing the below ground biomass, which is where we really want to hit it, is down in that rhizome. And so we're looking at about a 30 to 48% reduction in biomass, below ground biomass. That's in a 2-month period. So if we have a longer field season, we do expect that we're going to see better than that out in the field. So after all of this work, we did submit a petition for field release in April 2022. This went to both the US and Canadian federal agencies. Canada they have a much more streamlined, easier process than we do. So they already approved it in July of the same year. For us, we have a lot more steps to take. And it was recommended for release by an advisory group in July of the same year. And since that time, we have been sort of slowly plodding through the petitioning process with USDA, APHIS, plant protection and quarantine. So we are about to go into public comment. And when I say about to, I mean potentially in the next 6 months, going into public comment. And so after that, then we hope to get a finding of no significance, and then we can move forward towards releases. So in the meantime, we're starting to get ourselves geared up for preparing to do releases here in North America. So back in 2022, we started doing rearing in a quarantine facility. Since it's not approved, we can't put it on the ground, obviously. So we've got it in a quarantine in Sydney, Montana. And Natalie West is a researcher there, and her team is working on rearing techniques. The weevil's pretty challenging to rear, so we're trying to figure out the best ways to do it, so we have large numbers to put out into the field. CABI is continuing to rear and collect additional beetles, and they'll be sending those over to Canada and the US. They're also going to be assessing a pool rearing technique and doing some impact studies. Here in Puyallup and Montana, we are also putting in pools. I'm going to put in pools, and Montana is going to put in ponds. And we're going to assess potential, what's the best way to rear large numbers of weevils. So since Canada is ahead of the game and has it approved, they will be the first ones to get this out on the ground. I'm expecting that over here in the West, Hatzic Lake, which is in British Columbia, will receive the first releases probably this summer. Last year, this is Agriculture and Agri-Food Canada work. They did receive 26 weevils, but they didn't have any success with the rearing. The good thing is that the weevils live for multiple years and lay eggs for multiple years. And so they were able to recollect all of those weevils and put them into overwintering, and hopefully they will do a better job this spring. So they're going to start releasing in rearing tanks and also in Ottawa in isolated canals this year as well. So we are going to learn some things from Canada, hopefully. But in the meantime, we are trying to prepare ourselves for releases here in the States. And so we know that our initial releases will occur at Flathead Lake. This is where the population in the West is the largest. If you've ever seen that population, it's pretty mind-blowing. So we're starting to develop pre-release monitoring sites. And the reason we collect this pre-release monitoring data is it helps us understand what did the population of flowering rush look like before we released? And then we will release and we'll see how the population of the weed changes over time. And so this allows us to determine whether we're successful. And so this has been pretty challenging. We've done a couple of site visits. Aquatic systems are really complex, and I feel like every time we think we've come up with a plan, we find a reason why it might not work. So I think we've gotten there now, and this is all work being done with Melissa Maggio from the Montana Biocontrol Project, Bryce Christians with Missoula County Ecology and Extension, and then Natalie West, who is a federal researcher. So you can see that our pre-release planning days are really hard. We have no fun at all, but we are working out there trying to figure out what makes sense for these systems. And ultimately, we were able to come up with a monitoring plan. So if you're thinking, boy, flowering, I've got a lot of flowering rush and it's unlikely I'm going to be able to control it in some way, that other than biocontrol, I am looking for other potential sites, particularly here in Washington. So please do reach out to me. So all of this wouldn't be possible without funding from a really big consortium. We've had a lot of people involved, and really these biocontrol projects, when we have them, it's rarely that we have one big funding source. Instead, what we do is we pull funds from a variety of different, smaller amounts of funds from a variety of different sources, and then that helps make a project whole. So we're able to do a lot with little pots of money. And so we've had funding from federal agencies, the Kalispel tribe, and then multiple state agencies within Montana and Washington, and also from British Columbia. So from here, I'm always looking to be cultivating North American partnerships. How can we work together to really combat this really difficult weed? We're pursuing additional funding, particularly for that white smut, because I think it's going to be a really valuable biocontrol for us down the line if we can get something going after those fully submerged plants. And then, as I mentioned, we are trying to get our implementation plan figured out, and I would invite anybody to contact me if they would like to be involved. So with that, thank you very much.